In today's module, we're going to continue in the respiratory system, and we're going to focus on how the respiratory system adapts to exercise training in order to deliver enough oxygen to our muscles for aerobic metabolism generation of ATP. So the first slide that we're going to look at is looking at the rest to work transition. Right, this is going from sitting to a relatively light load, something below lactate threshold, which in an untrained subject would be below about 50% of VO2 max. So a relatively easy workload. So you'll see that the change in pulmonary ventilation observed in the transition from rest to a constant load is that the arterial tension, or the arterial PCO2, this is arterial here, I'll mark that, is relatively unchanged. We'll see a very slight dip in transition from rest to steady state, and this slight dip suggests that alveolar ventilation at the beginning of exercise is not as rapid as the increase in metabolism, right? So we have a greater usage of oxygen delivery and CO2 production from the metabolism, CO2 being produced from the carbon molecules in glucose, and oxygen consumed in that reaction. And so you can see more CO2, more oxygen being taken out, therefore less in the arterial, in the arteries. And so that slight transition does occur. However, we are able to meet our ventilation needs relatively quickly. The same pattern as before exists, right? So we reach steady state in ventilation if we're keeping the same absolute workload in about 2 to 3 minutes. Our rate of respiration, and therefore our expired ventilation per minute, that's the V dot E, which is in liters per minute, is going to reach steady state and then plateau and stay that for the duration of that low intensity exercise. Since we're in Texas, I figured we would talk very briefly about prolonged exercise in the heat environment. Again, here we're looking at PCO2 and ventilatory threshold. So we don't show PO2 because it will reflect almost exactly the same pattern as PCO2, just in the opposite way like we saw in the previous figure. So during prolonged submaximal exercise in a hot and humid environment, what we can see is that there's little change in PCO2 between the cool environment here in the white and the hot in the filled in, right? However, we can see down here that our ventilation is actually different. So if we're maintaining the submaximal load for an extended period of time, usually greater than 30 minutes, we will see that the rate of ventilation slowly creeps up, right? So again, thermoneutral environment here, not hot, not humid, or Tyler, Texas, hot and humid during the summer, right? So this increase in ventilation is due to the increase in body temperature, which indirectly is also an increase in blood temperature, which directly affects the respiratory control centers and will be the part of the next video. Because there's little difference in PCO2, this suggests that ventilation increases due in breathing frequency and dead space ventilation, and not actually PCO2 and O2 mediated. So let's look at incremental exercise, right? We've used this analogy as a walk, a jog, a run, and an all-out maximal sprint. And the first we're going to look at an untrained subject, and then the next slide we'll look at a trained individual. So what we see if we start here at the bottom is that our ventilation, our minute ventilation, or expired ventilation, as denoted by the V dot E, increases linearly, and we can see that here. So we see a nice little linear increase. This should say to about 50%. Sorry for the typo there. So we see a linear increase up to about 50% of VO2 max, where following that we see in general a change in slope, and we start to see an exponential or curved line above 50%. This exponential increase is an important point that we would denote as ventilatory threshold. which can be denoted as T with a V, with vent, right? Obviously, we can't use VT because VT is already taken by tidal volume, right? So this is this inflection point where we go from a linear increase of matching work rate with ventilation to then an exponential increase where we essentially, if you think about it like this, start to huff and puff, right? So as you work harder, therefore your rate of ventilation is exceeding your linear increase in work rate, and so we start to breathe much, much heavier. If we look at partial pressure of oxygen in the arteries, again, this is arterial PO2, you'll notice that for the most part, arterial PO2 in an untrained individual will remain relatively constant. We may see a slight drop, about 10 to 12 millimeters of mercury during very intense exercise, but as you can see, we don't have any problems in delivering oxygen to our tissue, right? Our arterial PO2 of 90, if we think back to our oxyhemoglobin saturation curves, that PO2 of 90 is going to be more than sufficient to cause a saturation 85% or greater without problems. And so we will be able to fully meet our oxygen demands by fully filling up hemoglobin here at these levels. One other point that I want to make, I'll see if I can draw a line here, is this incidence of ventilatory threshold and pH. So if we draw a line of pH in skeletal muscle, or sorry, pH in the blood and ventilatory threshold, one thing that you'll see is that these values line up beautifully. And indeed, if you remember back to earlier in the semester, one of the key things that happens in an untrained individual at about 50% of VO2 max is lactate threshold, right? So lactate is one of the molecules that helps increase pH, as well as hydrogen ions coming from metabolism and several other potential sources. But we see with heavy exercise, pH will start to slowly decrease, right? This change in pH will also affect ventilatory threshold, right? And indeed, this was interesting for a really long time. It was thought that this was a cause and effect relationship, and now maybe not so much, but it still is a great marker. So we can use ventilatory threshold as a very good marker as opposed to drawing blood to measure lactate as another marker of lactate threshold. So if we then look at an elite runner, right? So one thing that you'll notice, just like lactate threshold, that this linear portion here where this line exists for a much longer duration, right? So just like our lactate threshold, a trained individual's ventilatory threshold occurs at about 75% of VO2 max, and then beyond that, we then see our shift into our exponential huffing and puffing, as I like to call it, right? One other thing that you'll note is this elite, and by elite, I mean truly elite, we're talking competitive athletes, they actually will get some exercise-induced hypoxemia. So what this is arterial PO2, again, arterial here, arterial PO2 will see a stark decrease near 90 to 100% of VO2 max. So near maximal workloads, you can see that we are no longer getting large arterial PO2 values, which is telling us that if we think about the oxyhemoglobin curve, that we aren't fully saturating oxyhemoglobin and have a lack of oxygen delivery to the tissue. As you can imagine, that can definitely cause some effects in performance if you're unable to deliver that. So just note that this is an interesting case. There's lots of thoughts about it, and the current thought is that this doesn't even happen in everyone. Again, we're talking in elite, high-endurance trained athletes competitive athletes who are competing against the best in the world. So for most people, we'll just say that drops in PO2 aren't really going to have much effect on performance. We're not going to really see this unless you are truly an elite athlete. Some thoughts may be due to the ventilation perfusion mismatch, or remember this is our VQ mismatch. The idea is that we are perfusing, we have such a high rate of perfusion, right? So with these cardiac outputs in an elite trained athlete, I think we mentioned briefly in the cardiovascular section that it can be up to 40 liters per minute of cardiac output, whereas an untrained individual is 25. What that does is it says that the amount of blood going to the lungs is a huge volume, and it's occurring really rapid. So when it's really rapid, the red blood cell you can think of just zips through the capillary, doesn't spend enough time in capillaries to get sufficient diffusion of oxygen from the alveoli into the red blood cells, and therefore we are unable to have adequate exchange of oxygen in the alveoli and can cause this exercise-induced hypoxemia. And that's a great example of why we talk about VQ, ventilation perfusion mismatch, and a beautiful example of it in physiology. So in summary, at the onset of a constant load, submaximal exercise, less than 50% below lactate threshold, which would also, as we now know, be less than the ventilatory threshold, ventilation increases rapidly, reaches steady state in about 2 to 3 minutes, as we've seen in the other variables such as cardiac output and aerobic metabolism. Arterial PO2 and PCO2, while we see maybe a slight decrease, nothing truly significant, and are maintained relatively constant during this type of submaximal exercise. In a hot and humid environment, we can say our rate of ventilation drifts. We start to breathe more rapid rate due to body temperature affecting important respiratory control centers. And last but not least, we see incremental exercise going from rest to 100% all-out maximal exercise results in a linear increase in ventilatory threshold, about 50% in untrained to 75% or more in trained athletes. And at higher work rate, ventilation rises exponentially, right? And you've experienced this as again, huffing and puffing, especially really as you start to work much harder, your breathing rate just goes much, much higher, not in a linear rate. And this inflection point of when we go to a linear to an exponential increase is known as the ventilatory threshold. Next, we'll cover the cardiovascular control centers in the next video.